This is going to be part one of a two-part lesson in which we're going to look at the functioning of DNA. So you've learned about the structure of DNA, and now we're going to be talking about what DNA is doing in a cell. Just to remind you the structure of DNA, as we've discussed, DNA is composed of a strand of many smaller pieces called nucleotides. Each nucleotide is composed of a sugar called deoxyribose, a phosphate group, and one of these four nitrogen bases, either thymine, adenine, guanine, or cytosine, and each strand is held together by these hydrogen bonds represented in the center of the molecule. Now, it's those nucleotides and the sequence or the pattern of the nucleotides on DNA that actually contains the instructions, the code, to make a protein. It's like an instruction book for how to make a protein. That's what DNA is doing. It's all about proteins. So DNA is actually the instructions for proteins, and the proteins themselves are the product of the DNA. So it's the pattern of the nucleotides on the DNA, whether it's ATCG or CGAT, that is going to determine which protein gets made. So we think of it like this. We have the DNA molecule, and on the molecule we have genes, which we talked about. So this would be the strand, and this code, ACCAA, is going to determine the code on a molecule called mRNA, which we'll talk about in a minute. This process of creating mRNA from DNA is called transcription. And then the second part of the process is converting the message from mRNA into a protein, and this process is called translation. We'll talk about that next time. So let's start by talking just about transcription. To start with, transcription involves making this other molecule, it's another nucleic acid called RNA. And RNA's job is to carry the code out of the nucleus because DNA is not allowed to leave the nucleus. It cannot leave, it has to be protected within the nucleus so it won't be damaged. So instead, a copy is made onto this other molecule, RNA. Now, RNA stands for ribonucleic acid. Okay, DNA was deoxyribonucleic acid, so you can see it's very similar. It has three differences, however. It's a single strand, not a double helix. Secondly, it has this other sugar ribose instead of deoxyribose. And the third difference between RNA and DNA is that it has, instead of thymine, it has uracil. So when you're looking at the code, instead of thymine bonding with adenine as a base pair, instead we have uracil bonding with adenine as a base pair. Now, the RNA that carries the message out of the nucleus is the messenger RNA. We call it mRNA. There are two other kinds of RNA that are going to become important later. Transfer RNA or tRNA. Its job is to transport the amino acids. It's carrying them. I like to call it taxicab RNA, tRNA, taxi RNA. And then there's rRNA, which stands for ribosomal RNA, and it actually forms up the ribosome, which is the place where the proteins are made. It kind of makes the factory. So let's talk about the transcription process. The first step is that the DNA has to open up to be copied. It's unzipping. We use the word unzip because it kind of looks like a zipper when it opens. But basically, there's going to be enzymes that come and open up the DNA so that the base pairs are no longer paired up together. Then a special enzyme called RNA polymerase is going to start formation of the RNA. So you can see here, RNA polymerase enzyme has come and opened up the DNA, and it's going to start making a copy onto here, the messenger RNA. As the RNA is being made, the nucleotides from RNA are added according to base pairing rules. For instance, adenine, instead of with thymine, it's going to be paired with uracil, and guanine will be paired with cytosine. Cytosine is paired with guanine, and then thymine will be paired with adenine on the other side. So according to base pairing rules, you'll see this strand of RNA is going to be made from the DNA. It's going to pair up and just make the strand. So that's it for the process of transcription, and next time we will talk in more detail about translation, the second step where you take the code from the RNA and convert that into a protein, a sequence of amino acids.